Hi there, this is Rohan Mishra, and let us continue with our discussion that we have left. Okay, so this uh, in this lecture we would be talking about another important component in our drug discovery process, that is how do you obtain compound library and what are the steps that are to be followed when you try to process it. So for the sake of simplicity, we are going to study this process in 5 headings. First, I would let you know what are the sources of compound libraries, from where you can obtain those. That is, how can you create a compound collection. The next aspect that we are going to study about this compound library processing and preparation is that how do you clean up your compound collection, what are the filters that are to be implemented while cleaning your compound libraries, what are do's and don'ts when you apply those filters. Then we would also talk about how do we generate the 3D coordinates of the structures that are usually further used for virtual screening and molecular docking. We would also shed some light on the process like protonation, and we would also talk about some final filters that some people apply or they don't. This is how we are going to do. So here is what our protocol was, and we are already done with how do we identify the target protein. Then how do we obtain its structure. Either we obtain the structure from experimental databases like PDB, or we can then generate the model using homology modeling. After that, how do we identify the active site, and what are the other steps that are involved in processing your target protein, that we have already completed in my previous lectures. Okay, so in this lecture our main focus would be what to do with compounds and how do we perform how do we construct a typical compound library. Okay, so this lecture is for the ones who are usually working in bioinformatics or chemoinformatics field, wherein they start out with say some compound library which contains say some million compounds or say some few thousand compounds, and manually editing such compound library and processing such compound library becomes quite cumbersome. It is a very time consuming task, and therefore we require computational methods to get help in such compound library preparation and filtration. So if you are student from medicinal chemistry or organic chemistry, and you are doing your maybe PG dissertation, and you have synthesized a series of compound, say for example in range of 15 to 30 compounds, then it is not necessary that you follow all of this procedure. Okay, so what you can do is that you already might know what are the structure of your chemical compound. You can directly proceed for generation of the 3D coordinate of your structure, and directly proceed for the next step, that is virtual screening and molecular docking. So all in all, what I want to say now is ki, what if you are trying to explore some very new chemical space for a given target molecule. Okay, and if you have say some 10 millions or 1 millions of compound with you, how is that you should process, or how is that you should proceed, is the main focus of our today's lecture. Okay, so now to begin with, let us first try to discuss what might be the source of your compound collection, or where might I get compound library. So before diving into details of the sources of compound collections and their properties, well, let us for sake of simplicity divide your compounds into three major categories, or what you can say that whatever compound that you get, or whatever compound library you might acquire from different sources, they can be categorized into three major groups. Okay, so the very first major groups constitute called as reals. तो इसे अपन reals कहते हैं. So these group comprise a significant number of molecules, and these real contains marketed drugs and other known high activity compounds. तो जो कुछ भी marketed drug है, or the other compounds which are known to be highly active against a certain disease or certain receptor or certain target molecule, those are usually included in this class of set, or what we call it as reals. Okay, so these reals are thus a very valuable source, and those are usually routinely screened against a novel target, if to find out if they are active in a novel target as well. Okay, so this process is called as this process is called as drug repurposing, wherein you try to repurpose your drug and check if an existing drug can be repurposed to uh, treat some new disease or not. And usually most of the time things also work in this way. Okay, the next set of compound that you should know constitutes tangibles. So these tangibles are set of commercially available chemicals that you can buy from certain companies. So one can feasibly acquire them, or one can easily synthesize them in your own laboratory or in your own pharmaceutical company using well brought up chemistry that is already there. So tangible correspond to a set of compounds whose synthesis level is difficult, but somehow it can be managed if one have a good well brought up medicinal chemistry or organic chemistry laboratory at hand. The third category includes the virtuals. So these virtuals are extremely large set of molecule, that are very difficult to synthesize, and with whatever chemistry that we have, or whatever synthetic procedures that we have standardized so far, it becomes very difficult to synthesize this set of compound. So but it can be essentially be used as a resource for virtual screening in order to obtain novel chemical scaffolds that can be tried out by by synthesizing them, and maybe trying out on novel drug targets. Okay, so these are the three major types of compound. Those are usually given in pharmaceutical compound collections, like reals, tangibles, and virtuals. Okay, so now where can I get the compound, or where can I get the compound library? To answer this question, there is one-stop solution called as Chem Navigator database. So this Chem Navigator database is a web resource which correspond to the largest collection of virtuals and tangibles, and this database contains more than 21 million samples, and these 21 million samples represent about 13 million unique chemicals. So this is a very big resource for virtuals and tangibles. So this database is available on a subscription basis. So Chem Navigator offers compound for more than 154 companies, from which you can further or later on order them. So since it is available on subscription basis, you would have to pay some money to these Chem Navigator database curators. Sorry. So besides this, there are individual companies from where you can obtain those compound library. Well, say for example, there is a big company called as Acinix, and they provide a huge set of chemical libraries, which are also somehow categorized, or you can say specialized for a certain process. Well, say for example, they have got some 5,000 compounds that are targeted libraries for GPCR, G protein coupled receptor. There are certain set of compound libraries which are targeted for only kinase type of target. There are certain compound libraries that only target the ion channel or protease. So in this way, there are different companies. The web address of those companies are shown over here. The size of the compound library is also shown over here along with the description. I will share this PPT with you. Therefore, if you wish, you can further go and explore these company sites in order to download those compound libraries. So here is another slide which has got the same data. So here is your startup on where do I obtain my compound. Okay, so these are all commercial. You might or might not have to pay for it, pay for those compound libraries. Now the next question is that are there any resources from where I can get all those compounds for free? So the answer is yes. You can obtain the compound libraries from open source online resources like Drug Bank, like Cage Ligand, like you have got NCBS PubChem compound, which contains nearly a million or more than a few million compounds, which can be easily downloaded. There are also resources like ligand.info from where you can obtain such compound libraries. But since all these compound libraries are from public resources, well, that means either they are already published or they can be a proprietary component of, say, some pharmaceutical industry, or say some private organizations. So if you end up getting a very good activity of any compounds from such a source of compound library, it is always advised that you first check out if they have got any intellectual right protection or some sort of those things, so that you won't get trapped somewhere else when the drug discovery project proceeds further. Okay. So now, well, let us worry about what to do with that compound library that you have obtained either from that commercial source or from any open source resource as such. Okay, so let us start talking about how do we clean up the compound library. What are the steps that are needed to be carried out? uh, what exactly is needed to be cleaned up from your compound library. So whatever chemical vendors or whatever web resource from where you might download those compound library, well, these resources will give you compound library most probably in SDF format. SDF stand for structured data files. So it is one of the most popularly used file format for managing these compound libraries. So you might end up getting a compound library in form of an SDF file. So now there are many commercial as well as non-commercial compound collection that contain simple and small compounds like NaCl and waters that is associated with them. So the user need to spend a very significant amount of time as well as put a lot of efforts in cleaning up those collection, like removal of unwanted moieties, like removal of the solvent moieties, removal of salt moieties, and so on and so forth. Okay, so whatever compound collection it can be either virtual, reals, or tangibles. So once you acquire it from an external source, you would have to spend a lot of efforts for cleaning it up so that you can use in your further virtual screening experimentation. So Now, if a library is purchased from companies such as Chem Navigator, they might also provide their own solution to cleaning up and filtering your compound library. Okay, so there are other choices. Well, say for example, you can use softwares like Filter, which is provided by OpenEye. You can use Molecular Environment (MOE), or you can use Cytegic's pipeline pilot for performing such compound uh, I mean for cleaning up the initial cleaning up of your compound library. Okay, so now what exactly do we do in our compound library? Okay, so as I have already mentioned that some of the compound library might provide you with some compounds which are already associated with, say, some salts. So users is advised to split these covalent salt, and whatever the salt moiety is, he needs to remove it, and after removing whatever the main compound that has remained, he is supposed to normalize those charges. Okay, so how do we do such thing? How do we desalt your compound library? That is what the process is. The process is called as desalting, removal of salt and followed by normalizing whatever charge then it might have formed. So what happens is that almost all drug contain a bond between carbon and heteroatom. Okay, so the thing is, how can you identify in your compound library, okay, this is my salt and this is the compound that I am interested in. So in order to differentiate these two separate chemical moieties, what you can do is that you can identify which of the chemical component is salt and which of the chemical component is your drug molecule. So most of the time what happens is that if there are heteroatoms means what? The atoms which are other than carbon atom, these can be included in heteroatom. Okay, so what happens in case of drug molecule is that most of the heteroatom are directly connected to carbon. Okay, so we know that in a drug molecule, whatever heteroatoms would be there, those would be connected to your carbon atoms directly. So that would alternatively also mean if the compound is in complex with its salt, it would not contain heteroatom that are connected to carbon, but that heteroatom would be connected to another heteroatom. Okay, so if you write a kind of computer program that would identify the component from your compound library which contains heteroatom bond, then you have easily identified the salt molecules and thus you can remove them. Okay, so after desalting, the library is usually subjected to filtering. Okay, so you have obtained the compound library first, then you have made sure that there are no salts associated with your compounds, that is, you have removed the salt in the process of desalting. Well, followed by desalting, there are two ways through which one can proceed. Like, you can first implement the functional group filter that I am going to discuss with now, or eventually you can also implement the physicochemical filters first. So there are two type of filters that can be used for filtering your compound library. Okay, so these two categories are included as structure based, or what we call it as functional group filter, and the property based filter, or in other or in simpler words we can call them as physicochemical property filter. Okay, so let us first talk about what are these functional group filters, and then we would talk about your property based filters or physicochemical property filters. So it is not necessary that it is all up to you what kind of filter to apply first. Some people prefer to apply functional group filter first and then apply physicochemical filters, while some group tend to use the other way around. They tend to apply physicochemical filters first and structure or functional group filter later. Okay, so we need to discuss both of them. So well, let us start out discussing on what happens in functional group filters, or why do we need to filter our big compound library on the basis of these functional groups. Okay, so what happens in this functional group filter is that we here search for, say, some substructure uh, which are essential to be identified and discarded. Okay, because such functional group might bring out some undesired properties for your molecule. Okay, so what all in all I mean to say is that in this functional group filter, the substructure search is usually performed in order to identify and discuss and discard the compounds with known undesired groups in the library. Okay, so what now you need to do is that when you dissolve your library, you need to figure out if my compound library contains certain functional group that are already known to be associated with some undesirable properties for the given set of compound. Okay, so this technique can be applied to reactive functional groups, groups that are unlikely to be the lead molecules. Some functional group are always associated with promiscuous binders, and even functional groups that are classified as toxic. So these are certain class of functional groups that are associated with toxicity, that are associated with promiscuous binders, or that are associated with failure to form, or failure to generate a viable lead in further step of production of your compounds. Okay, so well, we need to identify such functional group, and we need to remove all those compounds which possess such functional groups. Okay, so now for a typical pharmaceutical screening, it is usually advised to remove all the compounds which has got some unwanted structures. So all of the compounds which has got certain functional group which we have termed them as unwanted structure, those compounds are needed to be removed. Okay, so the examples of such chemical substructure that can cause further interference with biochemical assays that are supposed to be performed through high throughput screening condition, well, these essentially include all these compounds. Well, say for example, you should always make sure that whatever compound collection that you have should not contain these leaving or hydrolyzable groups. So these leaving or hydrolyzable groups usually contains a functional group like sulfonyl halide, acyl halide, alkyl halide, right? Per haloketone, sulfonate esters, phosphonate esters, anhydride, aliphatic esters, halopyrimidine, imine, 1,2-dicarbonyl, aliphatic thioesters, epoxide, Michael acceptors, aziridine, so on and so forth. So these are known functional groups which are included in this hydrolyzable group. So compounds containing such hydrolyzable groups, functional groups, well, they do not perform well in further high throughput screening. Well, this is the knowledge that we have gained so far. So now, since we know that these functional groups are not very good for further high throughput screening and lead optimization or obtaining heat and further process, it is better to weed out all those compounds in the early stages of your compound filtration. So this is one of the group. Then it is also known that if there are compounds which contains direct heteroatom single bond that occur anywhere in molecule, so such functional group containing compounds are also not very good for further high throughput screening, therefore you need to remove them. So there are certain functional group that are known to be associated with cytotoxicity. So these groups specially include thiourea, aliphatic ketone, aldehydes, even cyclohexanone. Well, if such groups are present in your compounds or your compound library, they can potentially end up with certain compounds which are, cytotoxic in nature. So it is advisable to remove them in earlier stages of your protocol. Okay, so now, well, let us discuss that not all compounds that I have mentioned are always unwanted. One should always consider the term unwanted substructures in context. So I would like to cite an example. A large number of anti-neoplastic agents, that is anti-cancer agent, would be considered as reactive species according to whatever substructures I have shown now, because most of the anti-cancer compound might contain those functional group that I have already classified as either cytotoxic group, or they might contain heteroatom single bonds, or leaving or hydrolyzable groups. Okay, so furthermore, the second example that I would like to set is ki vast majority of flavor compounds are monofunctional aldehydes. Okay, they contain aldehyde group. So these are categorized into list of unwanted compounds that I have already mentioned before. So therefore, you need to always consider uh, the concept of unwanted functional group based on what exactly you are trying to perform. Okay, so therefore, when seeking activities in oncology, you might want to again revisit your definition of unwanted functional groups. So you might want to include some of the functional groups. Okay, so this unwanted stuff is supposed to be considered in context to what your objective of your compound library is. Okay, this is what I want to say. Okay, now your compound library might contain the chemicals with wrong chemistry. Okay, so what is wrong chemistry? A significant amount of wrong molecule might appear many times because of incorrectly drawn chiral centers. They might contain conformers, breach compounds, so on and so forth. So this can become a significant source of errors in further structure activity relationship studies, or when you want to build up a structure activity relationship model that you might obtain when you test those compound in bioassays, further bioassays. So now the visual inspection for all of the structure thus is not possible when the size of collection is really large in case of millions or say some few thousand compounds. Therefore, one has to use some automated procedure for detecting and perhaps correction of some of the wrong entries that might be present in your compound collection. So in order to do this task, in order to automate this task, there is one specialized software called as CHED, what we call it as CHED, and this is a specialized software that can be used for detecting what might be the wrong molecule that are present in your compound library, and how can we remove those wrong molecule. Okay. Alternatively, what you can do is that you can obtain whatever original structure file, usually in SDF format, that you have obtained. You can convert them into SMILES. SMILES stand for Simplified Molecular Input Line Entry System, using two or more conversion tools like Daylight Mol to SMILE, or you can even use open source softwares like Babel, through which you can convert your SDF file into your SMILES file format. So this is followed by canonicalization. Okay, and after canonicalization, you can check the resulting smile if it contains possible errors. So this can also be used as an alternative strategy. Okay, Then the next major issue that you should be worrying about when you deal with a very huge compound library is that it might contain duplicate compound. So this finding duplicate in screening library would significantly reduce the number of compounds that are to be tested. So this can be also be done by using smiles representation. What you can do is that you can generate unique smile, okay, so which are unique for each and every compound, and these unique smiles are generated by using atom ordering scheme, which is suitable for further molecular comparison. So once you have obtained the unique smile, you can compare those smiles with the database and make sure that your compound library don't contain any duplicate compound. Okay, so this is what I want to convey for this lecture. In next lecture, we would be dealing with the another aspect of bioavailability and physicochemical filters. Okay, so thank you.